I'm Dr. Nathaniel Chin, and you're listening to Dementia Matters, a podcast about Alzheimer's disease. Dementia Matters is a production of the Wisconsin Alzheimer's Disease Research Center. Our goal is to educate listeners on the latest news in Alzheimer's disease research and caregiver strategies. Thanks for joining us. Welcome back to Dementia Matters. While we primarily focus on Alzheimer's disease in this podcast, there are many different causes of dementia. Whether it be Lewy body disease or frontotemporal disease, there's so much we have yet to learn about these neurodegenerative conditions. However, there is one that we know very little about, known as LATE, L-A-T-E, or Limbic-predominant age-related TDP-43 encephalopathy. And this is a kind of dementia that causes problems with memory and thinking due to abnormal clusters of a protein called TDP-43. Yes, for those who recognize this protein, it is the same one that causes frontotemporal disease, but more on that later. Joining us to talk about LATE is Dr. David Wolk. Dr. Wolk is a professor of neurology and chief of the Division of Cognitive Neurology at the University of Pennsylvania Perelman School of Medicine. He also serves as the director of the Penn Alzheimer's Disease Research Center, co-director of the Penn Institute on Aging, and co-director of the Penn Memory Center. As part of his work, Dr. Wolk focuses on the diagnosis and care of individuals with a variety of neurodegenerative diseases and examining biomarkers that differentiate healthy aging from the earliest transition to Alzheimer's disease and other forms of dementia. Welcome to Dementia Matters, Dave.
Thank you. It's great to be here.
So I'm going to start with a basic question, but probably requires a little bit of time for you. And what exactly is LATE?
Yeah, so I think you said the mouthful well. So LATE is an acronym, Limbic-predominant age-related TDP-43 encephalopathy. Limbic-predominant because it affects generally memory areas and other limbic structures like the hippocampus and amygdala. Age-related because it tends to occur relatively late in life, more like in people's 8th decade, 9th decade of life. TDP-43 because that's the protein that builds up in LATE, and encephalopathy, which just is a generic term meaning it affects the brain. And it is really a very newly defined condition. It was in 2019 that the first pathologic criteria for LATE came out. And so we're just starting to learn a lot more about that condition.
So you use the word pathologic, which I'm glad you did, because for our listeners, my next question is how has it been identified so far? And now that we've identified it, do we know how common it is?
Yeah, so the vast majority of work with LATE has basically been done under the microscope looking at brain tissue, where it has been seen that in these older adults, people more than the age of 80 usually, although sometimes younger than that, and often in conjunction with other diseases like Alzheimer's disease, pathologists have seen under the microscope TDP-43 in these limbic regions, somewhat as a surprise in that, as you mentioned earlier, TDP-43 is often thought to be associated with frontotemporal dementia or ALS or Lou Gehrig's syndrome. And so the vast majority of studies have described TDP-43 as building up in these structures in older adults over time. And when it's been looked at in large populations, community samples or research samples, about 30-40% of people over the age of 80 will have this condition. So in many ways, it's actually a much, much more common condition than frontotemporal dementia or ALS as connected to this particular protein. More recently, there have been attempts to define the condition clinically in addition to pathologically. And so we're just starting to get a better sense of what it looks like in clinical populations.
And of course, when you say clinical populations, you mean living people. And so how are we discussing it, or how is it being planned to be diagnosed in living people? Because usually we're thinking about key features the same way we have thought about Alzheimer's. So what are the key features of LATE from a clinically diagnostic perspective?
Yeah, so I think, just in terms of taking a little bit of a step back, Alzheimer's disease historically was something that we definitively could make a diagnosis with at autopsy, whereas there were certain signs and features during life that helped us probabilistically get a sense of whether or not someone likely had underlying Alzheimer's disease. And I think we're kind of at the same stage as that with LATE. Now with Alzheimer's, we have all these great markers of the molecular pathology, the amyloid and tau that builds up in the brain. We don't yet have that for late. And so what you look for are features that are suggestive of this condition. And it turns out it overlaps quite a bit with Alzheimer's disease. And in fact, I think of it as one of the great mimics of Alzheimer's disease. Because it's affecting memory areas of the brain, it tends to be associated with significant amnesia. Unlike Alzheimer's disease, the amnesia tends to be a bit more isolated in individuals relative to other areas of thinking like language, executive functioning, visual spatial functioning. So it tends to be a bit more focused. It also tends to move a bit more slowly than Alzheimer's disease. There are some features that we look for on brain imaging, so on an MRI scan or on a FDG PET scan, which is a metabolic scan to measure brain activity. There are certain patterns that we see that are suggestive of this condition that might help differentiate it from Alzheimer's disease, including actually generally more severe hippocampal atrophy in this condition than Alzheimer's, even though we often think of Alzheimer's as being associated with hippocampal atrophy.
So if I'm a clinician and I have an older person, 80s, 90s, with only memory changes, and it's incredibly slow, there is a chance that this could be late. And you're working in this area of MRI imaging and FDG PET, which might be helpful differentiating that.
Yeah, so actually, I think one of the things that we've noticed in our own research cohort at University of Pennsylvania is that some of the cases that in the past we thought were Alzheimer's disease, but had always this it's really a little slower than what we would expect, have turned out to have late when we look at them pathologically. And so there are these features that are helpful in sort of increasing the likelihood that someone could have late, including the ones we described. We've also, though, been working on more formal criteria, clinical criteria that is about to come out. It's accepted, but will be published, I think, in the next week or two, where we also place an emphasis on ruling out Alzheimer's disease in these cases. And so in individuals who come in with a primarily amnestic presentation, who have significant atrophy, for example, in their hippocampus on MRI, and don't have markers of Alzheimer's, so they're amyloid negative, for example, most of those cases, many of those cases are going to have underlying late, particularly if they're in the right age range, in their 80s or late 70s, and certainly in their 90s.
And in a minute, I'm going to ask you a question about having both diseases and how that complicates things. But before, I'll take a step back and ask you, well, why does identifying and diagnosing late matter at all? Why should we care about detecting it clinically, just given that it's so new and there's no treatment?
Yeah, so I think there are a few reasons. One is that when patients come to see you, they want to know what their diagnosis is. And so I think, part of our job as clinicians is to kind of demystify what is going on with people so that they have a better sense of, what's happening in their brain, what's causing the symptoms they're having, as well as what might happen to them over time. And so one reason really that one would want to distinguish late from Alzheimer's is it has prognostic implications. So late does tend to be, again, a slower moving disease than Alzheimer's disease. Two, on the other side of the coin, we now have therapies for Alzheimer's disease with immune-based therapies where knowing that you have Alzheimer's is important as well. Three, I think because of those therapies, there are going to be a lot of patients that come into us with memory problems who end up not having amyloid evidence of Alzheimer's disease and aren't candidates for therapies like the anti-amyloid therapies that are currently approved. And those individuals, again, will want to know, well, okay, I don't have Alzheimer's, so then what do I have and what can I expect in the future? And then finally, once we start to be able to identify people during life as opposed to at autopsy, one can start to learn more richly about how the disease progresses, get more detailed testing and understanding, learn more about the genetics, and ultimately perform clinical trials in those individuals. And so you can't really test new drugs or things that are targeting this protein or this process without being able to identify those people in life. And so it's an important first step to even move on to getting therapies.
Well, then let's talk about the complicated mixed disease where you have multiple pathologies in the brain. How does co-pathology with late and other diseases for that matter impact Alzheimer's and Alzheimer's treatments like you just mentioned?
Yeah, so to me, I think this is one of the most important areas within Alzheimer's disease is that I think we all recognize now that probably the least common form of Alzheimer's is just Alzheimer's disease alone. And many patients, and up to 50% with Alzheimer's, have concomitant late pathology. There are other pathologies that are also common, like alpha-synuclein, which is often associated with Parkinson's and Lewy body disease. And it seems pretty clear from the data we have that when people have these co-pathologies, the disease tends to progress more rapidly over time and tends to have sort of mixed features of both Alzheimer's and these other conditions. And so understanding and knowing what the co-pathologies are in individuals who have Alzheimer's disease is, I think, critically important for understanding, again, what's going to happen with them, but also for us to begin to learn, do some of these new drugs that target Alzheimer's, how do they impact these other diseases? It could be the case that people with Alzheimer's and late maybe don't respond so well to these drugs. And so maybe in those cases, you might think, all right, well, this is not someone we want to prescribe a drug that, has potential side effects if it's not going to help. On the other hand, I think it's certainly possible, because there does seem to be a synergy between these pathologies, that maybe it helps with both conditions. And so maybe these are the patients that most robustly respond to these drugs. And so I think it's a really important thing for our field to begin to grapple with the heterogeneity of Alzheimer's disease, in particular related to these co-pathologies, because it really influences outcomes and ultimately will influence therapeutics. And hopefully when there are other therapeutics in the future that maybe target different aspects of all of these diseases, it might allow us to tailor our therapies to individual patients based on what the sort of conglomeration is of pathologies that they have.
So you're giving another reason why it's so important to study even our clinic patients, who are particularly those that are on these new therapies, so that we can better understand what's happening in the brain and how that impacts these outcomes.
Yeah, absolutely. I think as a director of our Alzheimer's Center, that one of the most important missions now that we have these therapies out there is to actually learn from what happens to patients with these therapies and to collect as much data as possible, obviously, with patients if they're willing to do so. Although I have found actually many of our patients are willing to share as much data as possible when they're on these therapies. And so we've built actually a program at Penn around this, where we're collecting research quality imaging data in all of these patients as they go through the course of therapies, banking blood for measuring genetics and proteins and other markers. We're banking spinal fluid if they happen to get a lumbar puncture, and adding in actually additional kinds of measures and tests, all with the idea that if we could start to learn based on some of the clues that we're getting about when individuals have these co-pathologies or other sort of sources of heterogeneity, how people respond to these drugs. And I think late, again, is an important player in this interaction with Alzheimer's disease and potentially the responsiveness to therapies.
One of the things I hear a lot when I'm out in the community is a phrase of, when you've seen one case of Alzheimer's, you've seen one case of Alzheimer's. Yeah. And while ultimately people progress through dementia and the changes that happen in dementia do feel similar as a clinician when you see multiple patients, it is a unique course. And a part of me does wonder, and I'm not asking you to answer the question, but a part of me wonders if that unique course is because not only a person is different, has different strengths and therefore different changes, but they could have different diseases in the brain that we just haven't been able to identify, such as late.
Yeah, I think I totally agree with you. And I think, trying to understand these co-pathologies may be one of the sort of sources of variance across patients that is going to help us better understand our patients. And clearly, I used to do a talk where I basically described like 3 different cases, all of them really completely different. And each of them were Alzheimer's disease. And the reality is that's not an atypical day for me in a clinic, is to see very varied patients. And I think a lot of it has to do with co-pathologies.
And you did mention, perhaps you can repeat it again, how frequently late occurs with Alzheimer's. But then do we know that is late occurring with Alzheimer's more than it occurs with vascular disease and Lewy body disease?
So it occurs by late stage Alzheimer's in about 50% of individuals will have TDP-43. Probably at more early stages, the numbers are a little bit lower, more like 20, 30%. And so when we think about people, perhaps when they would come to a therapeutic trial, maybe we're talking a little bit lower than 50%, but still certainly a large proportion of those patients. It occurs pretty frequently also, though, with Lewy body pathology as well. And I don't think we really know exactly the numbers relative to Alzheimer's Alzheimer's disease for Lewy body disease. There is some link with arteriosclerosis and late. So people who have small vessel disease are more likely to have late as well. And so there does seem to be an interaction with a variety of co-pathologies. And in fact, in Alzheimer's disease, when you have one co-pathology, you're more frequently likely to have yet another pathology than if you don't have a second pathology. So there does seem to be synergy across these different conditions.
I don't want to put you on the spot, but could you give us an example of someone who might be experiencing the symptoms of late, or how you thought it was Alzheimer's, it ended up being late, or vice versa? Just so that our audience knows, as a clinician who sees this, what should we expect? What does it look like?
Yeah, so I've seen a number of patients who have come in with, for example, mild cognitive impairment with primary memory issues. We'll do testing on them. And, when we do sort of standard tests, they'll show some problems in memory, often sort of modest problems in memory. Generally, though, pretty good function. Generally, other aspects of thinking are pretty good. And, you'll get an MRI scan and you'll see a little bit of atrophy in the medial temporal lobe. Sometimes, a little more than maybe you would typically see early in the course of, Alzheimer's disease. But generally, in the past, a lot of those patients we've just said, well, that seems like the early stages of Alzheimer's, they'll likely progress to a dementia level of impairment. And what we've noticed is they come in a year later and they still kind of look the same, little memory problems, still functioning well. And we're like, okay, it's slow moving Alzheimer's disease. And then they come in a year later and a year after that, and they're still showing kind of memory problems. Maybe the memory's getting a little bit worse and occasionally a little bit of language problems, but really not progressing at the rate that we expect to see for Alzheimer's disease. And you start getting a little suspicious about it. And then we've had cases like this where, we've since gone on and gotten, an amyloid scan or a lumbar puncture, and then they end up being amyloid negative. And suddenly we're like, wait, this patient doesn't have Alzheimer's disease. They have something else affecting their memory network. They have obvious injury to it. You can see it on a scan. And those cases tend to be the cases where late is this primary driver of their symptoms. And again, I think back to actually one of the first patients I saw, who I don't know to have late, but I really suspected. One of the first patients I saw at Penn who really had severe memory loss. I mean, constantly asking, why am I here? Why are we, doing this testing? And nothing else was impaired on our testing. And she came in for about 10 or 15 years. She passed away in her mid to late 90s and never manifested other symptoms of Alzheimer's. And I called her Alzheimer's all the way throughout the entire course and figured she had passed away from Alzheimer's. And again, we don't have autopsy on her, but now in retrospect, she probably had late. And, it was before we really even had that condition defined.
Well, you mentioned the value of, amyloid PET scans, spinal fluid, the future is blood-based biomarkers. Do we have a biomarker? Are we close to having a biomarker for late?
I think we're pretty close, actually. I think there's a couple of really promising lines of potential biomarkers for TDP-43 more generally, so not just for late, but potentially for other causes of diseases associated with TDP-43. There are something called cryptic peptides. It sounds kind of funky or crazy. They're mysterious, if you will. And basically, those are peptides that are produced in the setting of TDP-43 dysfunction, because TDP-43 plays a role in how RNA is processed. And so when it's not working well, you get proteins that you just generally don't see in normal health. While the protein levels aren't very high, there's a high signal to noise because there's not a lot of noise of those proteins in normal individuals. And so there's been some really compelling data that those may be really helpful for measuring this disease in spinal fluid and potentially even in blood. And then there's this other test looking at little vesicles, extracellular vesicles that bleb off of neurons and other brain cells that you can actually also measure in plasma. And they serve as cargoes for what's going on in brain cells that you can kind of measure in the blood. And there's been some really compelling data recently, much of it needs to be replicated, showing that you can pick up higher levels of TDP-43 in people who have conditions associated with it. And so I think between those two lines, I feel fairly confident that over the next few years, as the science advances and our detection advances, that we'll be able to have a biofluid fluid-based biomarker. There are also PET tracers that are in development that we'll also see as well. But one of my concerns with late is the amount of TDP in the brain is not so high that it might be hard to see that visually with a PET scan. But that also is a potential area that we could have a biomarker.
Well, that sounds exciting, and we won't hold you to the number of years. And you did answer my next question, which is what are the next steps or studies in the field? Is there anything else that you see in the next few years in the realm of late that you would want our listeners to know?
Yeah, so I think one will obviously be the development of better biomarkers, but I think we also need to do larger descriptive studies of these patients, as we do have clinical criteria out there that at least allow us to enrich populations in people who likely have this condition. And so I think we need to better sort of characterize those patients with regard to genetics, biology, but also just what their cognitive symptoms and other kinds of behavioral symptoms. I also think because there's so much work in targeting TDP-43 across frontotemporal dementia and ALS, that potentially some of the drugs that are being used in that domain could be applied to cases with late. And so I think we hopefully will enter relatively quickly a sort of therapeutic era where we're at least enrolling individuals in clinical trials. And so I would hope over the next few years that there are the beginnings of more clinical trials.
Well, that sounds exciting. All right, with that, I'd like to thank you for being on the show, and we certainly hope to have you back again.
Thank you. Anytime.
Thank you for listening to Dementia Matters. Follow us on Apple Podcasts, Spotify, or wherever you listen, or tell your smart speaker to play the Dementia Matters podcast. Please rate us on your favorite podcast app. It helps other people find our show and lets us know how we're doing. If you enjoy our show and want to support our work, consider making a gift to the Dementia Matters Fund through the UW Initiative to End Alzheimer's. All donations go toward outreach and production. Donate at the link in the description. Dementia Matters is brought to you by the Wisconsin Alzheimer's Disease Research Center at the University of Wisconsin-Madison. It receives funding from private, university, state, and national sources, including a grant from the National Institutes on Aging for Alzheimer's Disease Research. This episode of Dementia Matters was produced by Amy Lambright Murphy and Kaylin Rowerdink and edited by Eli Gadbury. Our musical jingle is Cases to Rest by Blue Dot Sessions. To learn more about the Wisconsin Alzheimer's Disease Research Center, check out our website at adrc.wisc.edu. That's adrc.wisc.edu. And follow us on Facebook and Twitter. If you have any questions or comments, email us at dementiamatters@medicine.wisc.edu. Thanks for listening.